Hello, everybody. It's Mr. Rogers here, and I'm going to talk to you today about carbon and the molecular diversity of life. And so this is I think, an interesting topic. This is actually a topic that is something that you cover in college, and it's something that takes a while. We usually call it organic chemistry in college, and you can take almost up to 3 years of it. And so there's a lot to learn in carbon and molecular diversity of life. And so we're going to kind of just hit the basics. We are going to learn a couple things that might be a little above and beyond what AP wants you to know, just because this is part of what I used to do as a pharmacology major. And so I just would like to use my knowledge. I paid a lot for it, might as well use it. So that being said, so although water makes up most of living cells, the rest are usually carbon. Carbon makes up most of the other weight. Carbon's a pretty spectacular element. When I was growing up as a kid, I always thought carbon was boring because it doesn't do as cool things as like oxygen and all those awesome halogens or those noble gases just have a sweet name and all those metals in the middle. But like, carbon is the most important element there for living things. All of our living things on Earth, every single one, not every single one uses water, but every single one uses carbon. There is also talk that maybe other places in the universe, they would use carbon or maybe different elements. Some have suggested the one underneath of it, silicon, as maybe a replacement for carbon for things. But as of right now, all living things that we've ever seen in this world are made from carbon as its backbone. So organic chemistry, like I said, I'm sorry about that, is the study of compounds that contain carbon. Organic compounds, as it says, range from really tiny little guys to like some organic compounds are visible by the naked eye as one compound. And almost all of these guys have carbons, and most of them also contain hydrogen in some form, either directly on the carbons or maybe on another atom that's connected to the carbon. So we'll skip that for now. I'm not really too worried about it. Here, these are some different organic molecules. These are the names for them and what they kind of look like. And so we talked about in the last presentation that carbon connects to 4 things. Hydrogen connects to 1. So these are some pretty straightforward guys here. You can see CH4 is 1 carbon and it's surrounded by 4 hydrogens. That's methane. Ethane is 2 carbons and 6 hydrogens. And ethene is a form of ethane with a double bond between the 2 carbons, so there's less hydrogens. So one of the things you can't do because you're online is honestly in person now, right now, is we can use ball models to kind of see what these guys would look like. And so one thing that's kind of hard to see in this PowerPoint, but I just want you guys to know, when something has 1 bond, they are a set distance apart. When it's a double bond, they get a little bit closer. And when it's a triple bond, they get even closer. The way to think about it is if I'm holding Mr. Urquhart's hand, we can kind of space ourselves out. But if I'm holding both of his hands in a very romantic way, we're a lot closer. And then if we hold hands and then put our shoes on top of each other, then we're even closer. So how many bonds an element can have in organic chemistry, discussed this in the last one too, depends on how many valence electrons it has on the outer ring. And you can kind of see here, hydrogen has 1, it needs 1 more electron, so therefore it is going to make 1 bond. Oxygen needs 2 more electrons, so it's going to make 2 bonds. Nitrogen needs 3 more electrons, so it's going to make 3 bonds. And carbon is got those 4. And you kind of see this is the Lewis dot structure here. You did those in chemistry, I think. You can kind of see here hydrogen would put another one. Well, you can put it anywhere in hydrogen. Technically, the dot's supposed to be there. But anyways, you kind of see oxygen needs a pal for this guy and this guy. Nitrogen needs 3 pals. Everybody needs a friend. So we can kind of predict what in organic chemistry, the main idea why it takes so long to learn is we can predict what everything's going to look like without even seeing it if you give me the actual chemical formula. So carbon dioxide, I know C, double bond to both O's. Urea, that's actually one of the ones that you need to make in your mole view. So it's on the next page, so I'm kind of giving it to you, but it looks like that. And so you can probably make a different combination with these things. And that's important too. Like you can get this same combination of things, but make something different. I always compare this in organic chemistry to like Mexican food. You really think about Mexican food, it's the same ingredients, just packaged in different ways. You can repackage this combination of things right here to make something besides urea. So these carbon chains are the skeletons of these organic molecules. The word is always backbone. We use backbone way too often. But you'll see in most organic molecules inside living things, they have a set of carbons here. Now, one of the drawings that we get down to like here, you will start seeing drawings that don't even show the carbons and hydrogens. And this is where kids start getting confused when they see chemicals. They're like, where the heck is everything? But in organic chemistry, our rule is pretty simple. We can draw lines. Where lines meet, that is where a carbon is. So there's a carbon here. And if I don't put anything on there, it's saying that there is a hydrogen attached to it that makes it its full set of bonds that it needs. So right here, it's getting a bond from this carbon and this carbon, so it needs two hydrogens. So if you look here, this is the same guy. There's bond, bond, two hydrogens. So you can kind of see, this is just easier to draw. Our scientists, you know, I wouldn't say we're lazy, we're efficient. And so instead of writing this out every time, it's just easier to just draw a little, I don't even know what that is, hexagon, makes it easier. Now, there might be other bonds in there too that kind of make things that crazy. This is benzene. This is the benzene ring, which is really super important in organic chemistry, especially making just saying making drugs sounds weird, but making pharmaceutical drugs, it's super important. Most drugs have benzene rings because they're very stable, and so we can deliver what we need to deliver. And so here, you kind of see it has two lines. So what that refers to is a double bond between these carbons. And because this carbon right here has two lines from this carbon and one line from this carbon, that means that only needs one hydrogen, and you can see one hydrogen. So you're going to see molecules with a picture like this, with no carbons. You're going to stare at them and be like, what's going on? That's what's going on. It's just, we're lazy. This is saying that, you know, these are full sets of carbons with whatever bond's not there. It's a hydrogen. So hydrocarbons are only carbon and hydrogen. These are hydrocarbons right here. These are chains. And depending on how long they are, makes them hydrophobic. And why they're actually hydrophobic is because really to make something be polar or to be hydrophilic, you need oxygens or nitrogens. So carbon and hydrogen are happy, they have no charge, so they don't like anything with a charge. And so this is a fat molecule here. This is what fat looks like inside of your human body. Now, with organic chemistry, there's also, like I said, different ways of creating with the same material. Structural isomers are setting them up differently, different arrangements. Geometric isomers have the same arrangement, but in different spatial arrangements, if that makes sense. So like picture my my desks in my classroom. If a different spatial arrangement would be my desk is in the same spot, but it's flipped upside down. And obviously that makes my class a lot different. Even though it's in the same exact place, a flipped desk is not the same thing. And enantiomers are isomers that mirror each other. So enantiomers are mirror images. The way we always teach it in college is enantiomers are your hands. You have a left hand and right hand. They are mirrors of each other. So you can put them to each other and you can see that they mirror each other, but if you put them over top each other, they are not the same. So if I put my right hand over top my left hand, I notice my thumbs are on opposite sides. I notice my fingers are not in the right spot. But if I put them together where my palms are touching, they match up. So those are mirror images. This is kind of a look here. Okay. So this is structural isomers. This is pentane, and this is 2-methylbutane. 2-methylbutane is what they put in lighters. They also put butane in it, but 2-methylbutane lasts longer. Structural, this is cis and trans. This is cis means same side. Okay, it means same side. Trans means opposite side. Obviously, these words have different meanings now in our common culture, but yeah, cis, same side, trans, different side. And then this is the mirror images, the enantiomers, left-handed, right-handed. Now, enantiomers are really important when making drugs. And it's really important in biology in general. For example, ibuprofen has a left-handed and right-handed. I know it says S. You would think it would say L. But S is the German or whatever the German word is for left. And so S ibuprofen works. R ibuprofen does not. And then albuterol, the right albuterol works. The S albuterol does not. Now, these are very, whatevers. We can actually separate these out for a long time. It wasn't a big deal. But what we've also learned is sometimes if you get the wrong one, if this is a right-handed thing, for example, glucose, if you get the wrong glucose, if you get the wrong side, maybe I'll have this as extra credit, but one of the hands, the right or left side of glucose, is actually poisonous to all living things. And so it's important to get the right one. If it doesn't fit, it doesn't fit. I like to think of it as we've all have gloves, and there are some molecules in the world that, like, for example, this ibuprofen, for ibuprofen to fit and for it to work, it needs to slide into a left-handed glove. And if I give it the right-handed ibuprofen, it doesn't fit into the glove, and it's ridiculous and doesn't work. That's the kind of idea. Now, there's other parts of your body where it's like a mitten, and it doesn't really matter if you put your right hand or left hand in the mitten, it can work. And those are for different drugs and different things. So it just depends on what's going on. So enantiomers are quite important. Oh, going backwards, sorry. We have the backbone of carbons. Now we can connect things to those backbones of carbons. And these are called functional groups. There are hundreds of functional groups, but there's a set ones that we're going to learn in this class. And each functional group is slightly different. It has different properties, and it changes the molecule and makes it unique. So, for example, this is estradiol. This is the precursor to estrogen right here. This is testosterone. You can kind of see that they're very similar, which kind of makes sense. You don't want a big difference between the two in the human body, but they obviously make a big difference. And so, for example, estradiol will end up making a female a female. Testosterone will end up making a male a male. So this is actually, both these are steroids. Steroids always have 4 rings. You're going to get used to this look of a steroid. And they have different functional groups. So testosterone has a double bonded O here, and estradiol has an alcohol. This is actually an alcohol group. OH is alcohol. Okay. This guy has a methane group coming off right here. Estradiol does not. There's also some other slight changes that are not functional group based. I mean, you can make an argument, I guess they are functional groups, but there's a benzene ring right here. We talked about benzene rings a couple seconds ago. At the bottom end of estradiol, there isn't one here. But you kind of see, we make slight differences, and they can make the world of difference inside of a biological thing. I don't think there's much arguing that this makes a huge impact on a human compared to each other. So there are 7 that we're going to learn about. There's hydroxyl groups, carbonyl groups, carboxyl groups, amino groups, sulfhydryl groups, phosphate groups, and methyl groups. Each one has its own unique property. And we'll talk about what it actually does in a second here. Okay? And so what I'd like us to do, I think I put them all on here. I didn't put them all on there. I'll get them all on there for you. I'll make a new activity. I would like us to learn, this is a flashcard assignment, I would like you to know these different functional groups and what is happening in each one. So I'm not going to go over each one here, but you can kind of get an idea. I will say this, is if anything has an oxygen or a nitrogen, about 99% of the time, it's polar. If something just has carbon and hydrogen, it's going to be nonpolar. So carboxyl here has an oxygen. This is a very, very polar thing. It's also acidic. And why it's acidic is this oxygen here will actually drop its H, just give it off, and give off an H+, and H+ is an acidic thing. H+ gives off, or it's what creates an acid. Amino group, again, has a nitrogen here, therefore it is polar. Sulfhydra, these guys aren't polar at all. These guys are pretty neutral, pretty basic. Or not basic, sorry. Long run, non-polar. Phosphates, look here. I would say that, yeah, that's pretty full of oxygens here. So this guy is polar. Methyl, it's a carbon and 3 hydrogens. It is nonpolar. So these different things are used in different situations. Now you can look here, say right here, we're looking at this chemical. We have this right here being nonpolar, but we have other things on this organic compound that make it polar. Basically this side right here. So when we're dealing with water, you would assume water would interact with this side, or this is the side that is used to interact with polar things. This side is used to interact with non-polar things. And so almost like having two keys, your body could use this in two different ways, or honestly a couple different ways. Like it could have a receptor that goes into here, it could have a receptor for here, it could have, we can use it in different ways, is my point here. So even with a functional group on something, other functional groups change what it can be used for on the other side sometimes. Now, I wasn't planning really going ATP too much because you guys, we do this later, but ATP is adenotriphosphate. Triphosphate. It's what we use as energy inside of our cells. It's the main chemical for that. There's other chemicals as well, but we simplify it here to just ATP. There's things like GTP and CTP and stuff like that. Let's see here. So that's used as energy. So reviewing things you should be able to know. One, explain how carbon's electron configuration explains its ability to form complex, diverse organic molecules. Well, that's because carbon connects to 4 different things. And so it can make different shapes and make a wide ranging of things. Like hydrogen, for example, hydrogen can only connect to one thing. So hydrogen doesn't have a lot of options. Carbon connect to 4 things, and then connect the one thing 2 times and connect the one thing 3 times. It has options, and so therefore it can create a diverse amount of things. Describe how carbon skeletons may vary and explain how this variation contributes to the diversity and complexity of organic molecules. Well, the more carbons and hydrogens you have, if we're just using a backbone of carbon and hydrogens, the more nonpolar that item is. Now, if I start adding functional groups to that, then I can get some diversity within that structure. I can throw some oxygens in there and make that substance polar. I can throw some nitrogens in there and make that substance polar. And then finally, distinguish among 3 types of isomers: structural, geometric, and enantiomers. Honestly, structural, we're just dealing with changing the shape. Geometric, we're kind of dealing with like where everything is. And then enantiomer, the left and right-handed thing. I'm going to be honest with you, number 3, we're not going to test or quiz over or anything like that. Number 3 is more of just a throw-in for you guys to kind of know at a deeper level. So 1 and 2 are important. If you have any questions, I'm going to have another video for you guys to learn about this, and also reading the book and also some of the activities. So we'll keep working on this. So I hope it's starting to make sense. If you need any help, let me know.